Today, we're going to start something new. And I actually really like the last two chapters. Like I say, the plan is for exam number 4 to be a week from today. So, much like before, it will be available all during class, all during the daytime. So, probably from like 6 or 7 a.m. until 11 o'clock at night. Um, and it'll follow the format of the exams we've seen here, a bunch of multiple choice questions. One thing I'm trying to make different that I'm actually playing around with now is I'm creating question groups where you need to choose, like, answer 3 of these 4 questions or choose 2 of these 4 questions and answer them. So if I can figure out the best way to code that, so people aren't getting zeros for the what questions they don't answer, um, they choose not to answer, um, I'll make sure that it happens that way. But that gives you a little bit more choice. And just because there's so much crazy stuff that we can go on with nucleic acid technology, it kind of saves your brain a little bit. All right. So um, let's talk about our new stuff, though. Okay. So these last 2 chapters, we're going to start taking all these kind of individual components. We've looked at proteins, we've looked at carbohydrates, we've looked at lipids, and now we've looked at nucleic acids. And we're going to start putting them all together and get our bigger picture of the cell as a whole entity, and then maybe even move on up a little bit to the organism. Although really, Biochem 2 is where we move on to organismal. We start looking at the interplay between different organs, how a whole organism regulates its energy consumption and output. Okay. But now we're going to look at our membranes and our transport. How do we get these proteins somewhere? Okay, we kind of saw that with our actin myosin walking along the filament there. See my invisible filament? All right, but we're going to look at this a little more broadly in here, and just kind of talk about how we put everything together. And in the next chapter, we start talking about signaling and communication as well. So I think and bearing with me, getting through the protein kinetics and going, woo, taking a breather with lipids and carbohydrates and then learning nucleic acids, we're starting to build. We're getting to the good stuff where we can really do stuff with it. All right. All right. So let's take a look here at our chapter 11. All right, so the big thing we're going to start with is just looking at our function and composition of membranes. And we'll even talk about a couple assays that are really cool and fun to say. Frap. We'll talk about the dynamics because, we tend to just think, hey, we have a big, membrane here and it just sits there. But it's very dynamic. There's a lot of changing that's going on, a lot of things that are happening. And depending on what happens to the organism, we can see big changes in there as well, too. Okay. We'll also talk about some membrane proteins, how our types of membrane proteins and how they interact with the membrane and what they can do. And then we'll talk about transport across the membrane and different transport mechanisms, from active transport to passive transport, primary, secondary, all sorts of good stuff coming in here. So I think this is an interesting chapter, and it's much less disjointed than the end of chapter 10. All right. So how do we make our membranes? If we go back and recall our lipids, our fatty acids, and our triacylglycerides, well, we got that glycerol up there. If we make a glycerophospholipid, we have this kind of polar head group and then this long hydrophobic tail. Okay, so we can see this with free fatty acids as well too. So our lipids are going to aggregate together due to the hydrophobic effect, and that polar head group is going to be the part that's going to interact with the water, and the lipids themselves are going to try and keep that on the outside and the hydrophobic part on the inside. So we can come up with some different structures here. We can get micelles, which we'll see here in a minute, bilayers, and then liposomes as well too. Okay, but we're going to concentrate probably mainly on bilayers, but we'll talk about micelles first. A lot of the structure will depend on what type of lipid we have. If we have a really long hydrophobic tail, we are going to make necessarily larger micelles. If we have a short tail, we can make smaller ones. Okay, and we'll also look at the concentration of these as well too. So let's talk about our micelles. We can kind of think of, say, here's our free fatty acid, okay, with our head group up here. we will have a hydrophilic portion up here. And then in yellow, we have our hydrophobic section in here. We essentially have this cone that kind of functions this way because our head group, that hydrophilic section, you know, has a greater sphere of influence, literally sphere. So we can take a bunch of these cones and put them together and we form a micelle. If you think of detergents for like washing your hands, you know, keep away the coronavirus and all that good stuff in there too, to get rid of dirt and grease. What happens is dirt and grease is typically hydrophobic. So when you wash your hands and lather up the soap, the detergent, the grease and dirt will get trapped on the inside of the micelle with the other hydrophobic particles, and then the hydrophilic head group interacts with water and gets washed away. Okay, so we can see stuff like this, sodium dodecyl sulfate. If you are bored in the shower and read your shampoo bottle, it probably has sodium lauryl sulfate in it. Again, these are all types of detergents that have this hydrophilic head group and hydrophobic tail that will help wash away dirt and oil. Okay. Just as a side note, if we looked at like first generation detergents that were made in like the 40s and 50s and 60s, they often didn't break down very well. You might go out in the streams and see like wastewater runoff and you'd see detergent bubbles there that would last for years and years and years because these detergents just wouldn't break down. Modern detergents are made to decompose a lot more quickly so they won't have such um, um, long lasting environmental effects. Anyway, back to our micelles. You know, depending on the size of our tail here and then the size of our wedge, if we have like a really short fat wedge, we don't need very many to fit together and we make a smaller micelle. So, you know, we might have a few dozen of these molecules. On the other hand, if we have longer tails in there, we might have a few thousand lipid molecules. Okay. So again, our concentration is dependent. Maybe another analogy of this is if you drop some olive oil on top of water and you can see the different droplets in there, depending on the concentration, you know, you might get, you know, couple big fat droplets or a bunch of small little droplets that are spread out all over the place. So that's maybe analogous to our aggregation in micelle formation for lipids present in an aqueous solution. All right. So what happens if we have a lot of lipids present? Okay, we can actually end up making a membrane bilayer. And here we're going to treat, this would be our typical representation of a glycerophospholipid. We have this large phosphate group, big honking negative charges up there, and we have those two tails that are hydrophobic. So we can treat this, especially since one of them most likely has a kink in it, we can treat this as more of a cylinder than a wedge. Okay, or they could still be wedge shaped, but our wedges are very close and we have so many of them that we can spread them out so that we can treat them effectively as cylinders. So when we do this, we can kind of stack them, one going one direction, one going the other direction, right? And we end up with a bilayer. And you can see here we have head groups on the outside and head groups on the inside. Okay, just arbitrarily referring to that. Hold on just one second. My dryer is going to continue to go on and off, on and off, on and off until somebody pushes the button. So I guess that's going to be me. I tell you. All right. Anyway, but again, what we have here are our phospholipids, our sphingolipids are both going to be excellent components for forming a lipid bilayer. Okay, so we've got the hydrophobic part on the inside. Whoops, didn't want to do that just yet. And our hydrophilic part on the outside. Okay, it's like an ice cream sandwich with the yummy fat-filled ice cream on the inside and the hydrophilic head groups on the outside. I guess that'd be the sandwich part. Okay, all righty. Okay, if we have enough of these, okay, besides making like a little sandwich, we can make a big sphere, okay, or a vesicle. So now what we have is the same thing, this lipid bilayer, but we've enclosed the center. So we have this aqueous cavity on the inside, and this vesicle can be suspended in a liquid solution, an aqueous solution as well too, but our interior cavity and our external solution are separated from each other. Okay? If we expand this out, we get a cell. We've got the outside of the cell and the inside of the cell, the cytosolic portion in there. Okay, all right. We can actually make synthetic vesicle membranes. We can take natural occurring vesicles and then flip them inside out to do stuff. There's a whole ton of stuff we can do with these. But a lot of our vesicles play transport roles. They can, and we'll see here later on that they can merge. Stop. All right. I don't know why my cursor disappears and won't come back, and then it'll just, yeah, anyway. Old technology here. I need to. Yeah, anyway, these can be good for transporting things, merging with the membrane and delivering, the contents maybe in that aqueous cavity into a cell. So we can see this for vesicle transport, and we can also see this in drug delivery models as well too. All right, and we'll see more about this in more detail when we talk about snaps and snares, probably on Tuesday-ish. All right. So, if we can arrange our, phospholipids or sphingolipids to make a membrane bilayer, but what exactly is a membrane? Okay. So one big thing is it's flexible. We've got pliable sheets in here. It's also complex because we have a lot of potential, phospholipids or sphingolipids that we can put in there, and we have proteins we can put in there, and then we can modify them and add stuff there. So we can make a very complex structure, right? Again, it's composed of lipids and proteins, occasional carbohydrates that are attached to either the lipids or the proteins. All of the cells have a cell membrane, and its main function is just to separate the inside from the outside. So you can think of it like the walls to your house, or all that good stuff in there. When we talk about transport, those will be like the doors or the windows that will allow you to come in or come out, okay? Or the chimney, because it's getting close to Christmas, right? No, too early. All right. Our bacterial and prokaryotic organisms tend to just have like one big inside versus outside, not a lot of internal organelles. But our eukaryotic cells, we have our nucleus, we have our mitochondria. If you're a plant, you have chloroplast, you got your various ribosomes, Golgi apparatus, all sorts of internal compartments in here that will have their own membranes that will separate, say like the mitochondrial section from the cytosol inside the cell. So we might think of this analogy, our eukaryote, as like a big apartment building, and each of the organelles is an individual apartment in there. All right. If we take a quick look at an electron micrograph of a membrane, you can see that our membranes actually show up as being relatively dark in here. Okay. Maybe if I can get my cursor to show up. Good Lord. I've got to get an external mouse. But this is our endoplasmic reticulum. Our membranes often will have proteins associated with them, so they tend to be a little more electron dense, which is why they show up fairly well this way. You can see our mitochondria tend to be pretty dark because they are packed with proteins, and our secretory granules are going to be really dark because we have stuff in there that we're getting ready to deliver out someplace. And you can see our nuclear membrane. So this is just an electron micrograph showing us, that there are tons of membranes associated within a eukaryotic cell here. Okay. All right. So we already know the major function of membranes is to separate the inside from the outside. Okay. But just like if you build a house, you got to have doors in it, doors and or windows. So you got to allow things in and out. So our membranes are going to contain gates or pores or transport apparati that allow the import and export of nutrients or waste materials. Okay, so we want to bring in food and take out the garbage. All right, kind of like what I did last night. All right, retain metabolites and ions with the cells. So important things, if we make a lot of ATP for energy, we want to keep that in the cell so we can use it in there. Our membranes also need to sense their surroundings. Again, living organisms sense their surroundings. So we may have external signals, and we can transmit that information into the nucleus and say, hey, turn on this gene, or maybe to our cilia and move towards food or move away from the poison. We'll also see that compartmentalization in there. One of the important things, and this plays a major role in metabolism, is we separate energy producing reactions from the energy consuming ones. If we are making ATP, we don't want to immediately burn that ATP and just make AMP and then convert that back in. So we don't have this futile cycle. We'll use a membrane to say, hey, we're going to make the ATP over here, and then we'll transport it over here where we can utilize it for energy, and then take that AMP and back and remake ATP. Right? We transmit signals, store energy as a proton gradient. Oh, I love the electron transport chain and the mitochondria. It's awesome. And we'll support synthesis of ATP, again, our energy currency of the cell. Okay. So membranes have a lot of common features. I'm not going to be super particular about the measurements in here, but we're typically, most membranes are somewhere around 60 angstroms thick in there. It just works out that that's about the appropriate length. If you think of our phosphoglycerides, and our palmitic acid being one of the most common ones, is about 16 carbon chain in there. So if you have two of those with the head groups, it just works out that that's somewhere around 60 to 65 angstroms. So depending on what substituents we have in our lipid bilayer, that can be a little bit thinner or a little bit thicker. Okay, and if we add other stuff in there too, like the question on the previous exam that I think just about everybody got right, what happens if you insert a cholesterol into a membrane? It thickens it, makes it more rigid. So we can see that in there. Okay. Main structure of membranes is a lipid bilayer. Okay. Now there are some weird archaeobacteria out there, and I think we may have seen, I don't know if that's in this chapter, it's one of the early chapters. There's one that essentially makes rocket fuel, and instead of having a lipid bilayer, it has a lipid monolayer, but it has this long hydrophobic dual chain in there, and two, like a head group and a tail group, that are both hydrophilic, so that it functions as the membrane. But unlike a lipid bilayer, which can be separated apart, you heat it up enough, or treat it harshly enough, it'll separate. This monolayer can't separate, so it essentially holds the, I think it's dihydrazine, essentially rocket fuel that these bacteria are making, but it keeps it compartmentalized. So membranes will spontaneously form. It's energetically favorable for them to form, because if we have lots of hydrophobic components in there, our hydrophobic effect, if those hydrophobic components coalesce, that is energetically favorable, because it allows more freedom of rotation for water molecules in there. Okay. We'll often see that our membranes contain proteins, and many of these proteins will span the lipid bilayer. Not all of them. Some of them are loosely associated, and some of them are just kind of on the surface there. Another important feature of membranes, they're asymmetric. Okay, the outside layer is going to have a different composition than the inside layer. Okay, and if they flip over, they can flip. Usually, that's an indication of some major trauma or issue that needs taken care of. The other neat thing is, I mentioned we can attach carbohydrates. We only find carbohydrates on the outside layer, never on the inside, and that has to do more with like signal recognition. Once you're on the inside, that's the cell, that's the organism there. If you're on the outside, then we can use that as a pattern, or a binding moiety, something like that. Another interesting fact, our membranes can be electrically polarized. We can have an electric potential across the membrane, so we can actually have this charge potential, and we can use electromagnetic energy as part of this. We can think this, if we're pumping ions across, we're pumping charges across, we keep them separated, we might have a more negative charge on the outside and a more positive charge on the inside. So you're going to have a desire for current to flow there, which can be harnessed for energy. Okay. So if you go back to like the original Matrix movie, he's like, you use humans as batteries. Well, technically, yeah, you can in a way. Okay. Not really good batteries, but well. Anyway, and again, our membranes are relatively fluid. Okay, we'll talk about this. They're like, chunky fluids, viscous, right? So this leads us into something, and this probably seems like a terribly long time ago for you guys, but for me, it's like 1972. It's like, well, it's not that long. That's not that long ago for me, because I'm old. But the fluid mosaic model of membranes is relatively new, and the idea here is that these lipids are making this really kind of thick two-dimensional solvent that can move around. It kind of stays together, and within the membrane, you'll have rafts. Oh no, I need more better stuff here. There's actually some nice little videos I think I've got linked in here that will show that. But you have like a little raft area that is associated with a particular process, and those can move along the surface of the membrane here. Um, the idea too that proteins can either be embedded in the membrane or they can just be associated with it kind of floating on top. All right. So it's kind of like when you go swimming, you're down in there, but you could wear like those funky walk on water boots and be on top of the water as well too, I guess, or go water skiing or something. There's a better analogy out there somewhere. Um, so when we talk about these proteins, integral proteins are the ones that are firmly associated with the membrane. They are in the membrane. Okay. Peripheral proteins are the ones that are just weakly associated. They might be just bound by by a non-covalent interaction. Just a charge or hydrogen bonds holding them in there. Some of them will be covalently bound, and we'll see GPI, glycerophosphoinositol, I believe, is the um, lipid that they're commonly bound to, but it's just a single linkage there. So you can find an enzyme that will cleave that linkage and separate that peripheral protein fairly easily. Okay, so here's our picture worth 1,000 words. Okay, we have our membrane bilayer in here. Most of the time, we're going to talk about phospholipids, so we kind of draw those in green. In blue. In green, we might have our sphingolipids. Again, you notice our tails look very similar. Okay, so very, very, very similar, just subtle differences in here. Our little orange bits that are in there, I believe, are our cholesterol representation. Come on, cursor. There we go. Okay. All right. Yeah, sterols, cholesterols. Here we have an integral membrane that has a single transmembrane spanning region. We're going to talk about this a little bit more, but here we have this alpha helix that essentially, is going to be composed of maybe alanines and leucines, form really good alpha helices. They are also hydrophobic residues, so they will fit in there very nicely. And then, our outside and inside portions will be more polar. All right. We can see oligosaccharides that are attached, so we can make our glycoproteins. We can also attach our carbohydrates directly to a lipid, so we can have a glycolipid that way. And then here we have a protein that has this little sugar moiety that connects it to a phosphoinositol in the membrane there. So different ways that we can connect them together. Okay. We can see in here we have a peripheral protein that is linked to like one little lipid tail. So it's basically got like one little anchor in there. Okay, but that's pretty easy to kind of pull that out as opposed to trying to get this whole thing out that's in there. Here we have a peripheral protein that is just loosely associated with the head group. So it's probably hydrophobic effects in there. Okay. All right, so it's a big picture, a lot. What we have these drawings in here might represent pores or transport systems that allow us to take a molecule from, say, the outside and bring it to the inside, or vice versa. Okay. All right. So, how do we know what our membranes are made of? And why is it important? Okay. So our membranes are going to vary by different organisms. So your dog cell protein is going to be different than your cat cell protein or something like that. Even our different tissues, your liver and your heart, are going to have different compositions in there, and even the organelles within are going to have different compositions. So your Golgi apparatus is going to be different from your mitochondria, and so on. We'll also see that there will be a variance in the ratio of lipid to protein. I'm a mitochondrial biochemist by training, so I love mitochondria because they are packed with proteins. I think proteins are the most interesting part, and they're just packed with protein. Super high ratio of protein to lipid. On the other hand, things like storage vesicles might have just be almost like 90-95% lipid and very little protein in there, just enough to interact or signal um, signal recognition. All right. But the type of phospholipid is going to vary. We can also look at our sterols, our various cholesterols. Those concentrations and types will vary as well too. Again, most of our prokaryotes, the vast majority, lack any sterols at all. There's a rare few that have a couple in there. But for our eukaryotes, we're going to see the cholesterol is primarily found in the plasma membrane, but there's almost no cholesterol in mitochondria. So again, healthy to eat. No, okay, no, I'm kidding. And in plant chloroplast, we'll have these galactolipids, but for animals, almost no galactolipids present at all. Okay. All right, so we can look at compositions in here. This is just an example. I'm not going to ask you guys to memorize this or know anything like this, but if we look at our components by weight, we can see that the myelin sheath insulating the nerve cells is 30% protein, 30% phospholipid, and 20% cholesterol, something like that. Compare that to, say, the mouse liver, we get a lot more protein and a lot more cholesterol, about the same amount in here. It's like the other stuff that's in there, galactolipids, plasmalogens, so on. You can compare this to E. coli, 75% protein, 25% phospholipid, no sterols. Okay. Paramecium, yeast, and it just on, so our components vary by our organism. Right, that's all we're really saying here. All right. And our different organelles, okay? So you can look at your plasma membrane, your intermitochondrial, your outer mitochondrial membrane, your lysosomes, your nuclear membrane, your endoplasmic reticulum, and your Golgi apparatus. You can see that we have different amounts of cholesterol. Maybe, there we go. Maybe I just don't have much of a soul and my trackpad can't figure out that I'm touching it. Okay, but you see the plasma membrane, tons of cholesterol in there, okay? And you go to, say, the mitochondrial membrane, very little cholesterol. But we do see a lot more cardiolipin, same thing in our lysosome and our smooth ER, a lot of cardiolipin in there. Sphingolipids, lysosomes have a lot of sphingolipids, mitochondria, very few, but not so much in there. Phosphatidylcholine and phosphatidylethanolamine are both very common, but you can see that we vary their percentages across there as well too, and then there are other things there as well too. Okay, so again, this is just kind of a rough map of the percentage composition of the different types of lipids for the different membranes of various organelles. And I believe, okay, yeah, this is rat hepatocytes, so we're looking at rat kidney cells here. All right. So the other thing that we mentioned is that there is asymmetry. So the outside and the inside has a different composition. Okay. So if I look at the trans Golgi apparatus in here, you can see that I'm going to have a lot more sphingolipids on the outside and some cholesterol here that's kind of associated to kind of pucker this up. And on the inside, I have a lot more phosphatidylinositol, and phosphatidylserine, and what's the other one in here? A little bit of phosphatidylcholine, but primarily phosphatidylcholine is on the outside, and these are on the inside. Okay. If I look at the endoplasmic reticulum, I can see that it's a lot closer. There's not as much variance. We see a lot of the same type of stuff going on there as well, on either side. Okay, but the Golgi apparatus very different from the inside to the outside. All right. So this just leads to the idea that our membrane bilayers are generally fairly asymmetric. We got different compositions on the inside and the outside. So it's like maybe in your house you have a brick facade on the outside and wooden walls on the inside. All right. Yeah. So that's my rough analogy there. Typically, the outer leaflet will be more positively charged. The inner leaflet will be more negatively charged. So we can have our electric potential across there. We also end up seeing phosphatidylserine. So we have different components that can play some pretty significant roles. So phosphatidylserine is normally found on the inside. For platelets, if it moves to the outside layer, it says, hey, there's damage. We're going to activate blood clotting. Okay, so that's what it does in platelets. On the other hand, phosphatidylserine, it's found in other cell types as well too. If it moves to the outside, it's the kill me now sequence. Phosphatidylserine on the outside says, okay, time to die. For platelets, though, it says time to clot. Okay, so when we, depending on the composition, even if it's the same glycerophospholipid, it can have a different meaning depending on the type of cell. All right. I'm briefly going to mention archaebacteria because, again, these are the weird stuff. All right. They often have some unusual chirality. All right. Typically, we see D-glycerol for bacteria, but we have L-glycerol that gets used in archaebacteria. I had that question about D-glucose versus L-glucose. What happens if we ingest the wrong one? I'm like, ooh, I can see who has the Lippincott's Illustrated Review, because I think I stole that question from one of the versions of Lippincott's Illustrated Reviews in there, but it certainly won't be incorporated into any long chains, but it can still be broken down, it can still be metabolized for energy. Okay. But we can see archaebacteria use the wrong version compared to everything else. They'll often have some really unique fatty acids that make up their constituents. Again, you think like some of these archaebacteria are the things like the bacteria that grow in the volcanic vents where it's really high temperature. So they may need branch chains or something funky in there to help them stay together. They may have ether linkages instead of ester linkages. And again, some of them have a monolayer, like the one that makes hydrazine. Here we go. Yep. This is one of our volcanic hot springs bacteria. At this high temperature, that would be enough energy to separate a membrane bilayer and just kind of cause it to melt apart. So instead of having a bilayer, they have this monolayer where you have glycerol on either side, but these diphenyl groups that connect those two and basically function as a long monolayer. So it's the same width, it just can't be melted apart because it's a monolayer instead of a bilayer. Feel like I drank my caffeine a little too close to class time. Probably going super fast, aren't I? Reminds me, let's do a little time check here. Okay, we're good. We're where I want to be. Okay. So, besides having our phospholipid and sphingolipid and cholesterol or sterol derivatives in the membrane, we've got to make it more complex than that. And we can have different types of lipids, but let's throw in some proteins where we have a huge amount of variety. So our proteins and membranes can play a huge number of roles. They may function as receptors. So they can detect signals like light. Light counts as a signal, so we get opsin for our eyes. We have our hormone receptors, insulin receptors, retinoid X receptor, stuff like that. Neurotransmitters. So we'll take a look at neurosignaling coming up here in a bit. We're going to see the acetylcholine receptor that's going to bind to a protein, and then that's going to cause endocytosis to bring the signal inside. Pheromones. So our taste and smell receptors in there. We also have all these sorts of channels, gates, and pumps. So we can have nutrients, maltose gets across through maltoporin, ions, our K channels, our potassium channel, and again, neurotransmitters or serotonin reuptake protein. So I remember watching TV years ago and some antidepressant drug, I forget what it was. These ads don't say, but they had a cool animation. They had the what was it? Serotonin reuptake. I can't remember if it was a happy or sad pill, but the serotonin would try and get in, it couldn't get in. So, it's like, oh no. And then the drug would allow the serotonin to come in. It's like, oh yay, happy. So they had this nice little animation in there. But our serotonin reuptake protein and serotonin blockers, and maybe it was the blocking it that made it happy. I can't remember. Bad ad. Sort of good ad, but bad ad because it doesn't stick with me. We also have, of course, enzymes in there as well, too. So we can look at making more lipids. We're altering our lipid composition, our acyltransferases. We can look at our ATP synthesis in there as well, too. And we'll see a picture of that as well. All right. So let's break our membrane proteins down into three groups. Okay. So we'll start with our peripheral proteins. Okay. And these are ones that are loosely associated. Okay. They're often bound just by a hydrophobic interaction, or sometimes by a hydrogen bonds as well. Okay, so they're easily dissociated. If we change the pH or change the ionic strength, this protein will just fall right off. So very easy to purify. Okay, we can also have amphitrophic or GPI linked proteins. They're linked at particular times. All right, so maybe they have a little hydrophobic tail that unfolds and integrates into the membrane, and then we can change the conditions so that it goes away, or we can have this anchor in here with a Um, Proteoglycan, we can use phospholipase C to cleave this sugar chain, and we can free that away. So it's just like one little tether. You think of like a like a hot air balloon. They often have all those ropes. So this is a hot air balloon with just one rope. So you walk up with a big knife and it flies away. I don't know. It's an analogy I come up with. Okay, but these are more strongly associated than our peripheral proteins. So we may need like one little enzyme reaction or something to change in there. But if we trigger whatever regulatory event is there, we can cleave that off and separate it fairly easily. Okay. On the other hand, our integral proteins have a large hydrophobic region. Okay, you might have one that's partially in there, so it's like half of it is hydrophobic and half is hydrophilic. Or what I tend to think of more commonly, you have an internal and external portions, the inside and outside, the parts that are sticking out into the cytosol or the extracellular area is hydrophilic, and then the yellow is hydrophobic and is transmembrane that way. So they're a lot harder to get out. You can use strong detergents. Our detergents can kind of function as a surrogate membrane to bind to the um, hydrophobic portions of the protein and help separate it out that way. All right. One of the issues we had when I was in grad school was trying to crystallize the iron sulfur protein. And the iron sulfur protein is a massive transmembrane protein with a huge region that is transmembrane. I think it had something like a dozen alpha helices that crossed there. So it had a lot of hydrophobic character. So if you tried to put this in solution to get a crystal formation, it just wouldn't work. So the workaround was to use an antibody that bound to it, and the antibody had a huge region on it that was water soluble, so it kind of overwhelm. You have like your tiny by comparison um, um, complex 3 and this huge antibody fragment, and you could crystallize the antibody fragment and it would just kind of hold that hydrophobic section in there on its own. So a fairly complex crystallization procedure. All right, so again, peripheral ones are just loosely associated with the polar head groups, very loosely associated. Change the pH, change the salt concentration, and they fall right off. Okay, they're the bandwagon supporters. It's like, oh yay, my team is doing really good. I'm going to go cheer for them. It's like, oh, they lost 3 in a row. Yeah, I'll go do something else. All right, our amphitrophic are the ones that are loose, that are associated, but we can remove those under certain conditions. We have a regulatory function there. We can either like cleave off a binding site with phospholipase C, or we can have phosphoinositol, remove that, and our protein falls off. Okay. Um, our lipid linked ones, we can have our nice little anchors in here as well too. So maybe we have a lipid anchor. So here we have our protein. We have this sugar moiety that functions as a bridge to a phosphoinositol, and there's our lipid anchor in there that kind of goes into the membrane. It's a terrible picture. They should probably be beside each other because this is really glycerol in here, right? Okay. But again, we have these nice little anchors in here, whether it's maybe a palmitoyl group or a myristoylation or farnesylation in here. So we have a farnesyl group that can be associated in there as well too. And again, we can cleave these and separate these out pretty well. Okay, so they're anchors. We call them anchors because it's much like the anchor on a boat. You can even have like a sea anchor. You kind of throw it in there and it kind of keeps you from drifting too far, but it doesn't keep you in one place. If we think of that in terms of our lipid raft, our dynamic membrane, that might be a good analogy in there. They're typically going to have this covalently linked lipid molecule, whether it's phosphatidylinositol with some type of carbohydrate group on it, or sterol or long chain fatty acid in there. That basically that lipid part becomes part of the membrane and functions as an anchor. You can cleave off the anchor and free the membrane. Okay. The book likes to talk about farnesylation a little bit. I rarely see it, but it's well studied, it's well known, so it gives a good example of how this actually functions. We can actually target a particular protein to the inner leaflet of a plasma membrane by farnesylation. Talking about carbohydrates, we only find those on the outside, while farnesylated proteins are only going to bind to the inside in here. So a lot of times if we want to get a protein to the membrane and then work on it there, farnesylation can be a good intermediate. It's going to take it to the plasma membrane on the inside, and then we can do something else to it. Okay. Um, the neat thing about this is we have identified the sequence of the protein um, that uh, makes it a target for farnesylation. It's basically the CAAX. Okay, so we have this conserved cysteine followed by two aliphatic amino acids, followed by either methionine, serine, glutamate, or alanine. I just don't seem to have a lot in common, but that's what we've limited it down to. So if we have a sequence like that, then farnesyl transferase will recognize that sequence and will add a farnesyl group to the protein as an anchor. Okay, so farnesylated proteins go to the inner membrane and collect $200. Non-farnesylated proteins go directly to jail and do not collect it. Okay, a little monopoly joke there. I don't know, but non-farnesylated proteins don't go to the membrane and they're inactive. One of the neat things about this is when we start looking at oncogenics and cancer therapies, um, how we can get a protein to be active or inactive becomes a potential um, uh, treatment for various cancers to activate or inactivate our cells. So we can get rid of the farnesylating agents or the sequence in there or something. We may have another method of treating certain types of cancers. All right. So the last type of protein are integral membrane proteins. And again, just kind of blowing up that picture, these are things that are going to be either deeply inserted or transmembrane, go all the way across in here. Okay. They have a lot of asymmetry like the membrane. So, we're going to see, parts that are, you know, peculiar to the inside or the outside. They're usually not going to be the same thing on either side in here. We'll see these long hydrophobic stretches that are in here that will allow it to interact with the interior lipids, the tails, the hydrophobic tails of the phospholipids or sphingolipids in there. If we disrupt the membrane with detergents, we can also remove these proteins as well too. The only trouble is if we purify these, we often bring the detergent with us. It's hard to get rid of the detergent because we got to have some type of something there to um, associate with that hydrophobic effect. All right. Okay, so we draw this cartoon here and say, yeah, we just have this big greasy region in the middle, right? But that greasy region is actually probably not all contiguous. In this case here, we have a seven transmembrane region of alpha helices. And then, our connections between these alpha helices are probably going to be hydrophobic on either side, whether it's the outside or the inside. But we have these nice little hydrophobic runs that will be transmembrane and anchor this protein into the membrane. Okay. So we can see something neat about this as well too. If we look at charged residues that are in blue, we see these charged residues primarily either on the inside or the outside. So here we have potassium channel, multiporin, OMPLA, OMPX, phosphophorin, all sorts of things in here. The vast majority of our charged residues are on the inside or the outside. Now, occasionally you see some charged residues on the inside in the hydrophobic region here. And typically those are going to be conserved residues because those are going to play a role in the transport mechanism. All right. So if there is a charged amino acid somewhere inside the lipid bilayer, it's probably part of the mechanism for how the transport protein functions. Okay, maybe it stabilizes a charge as you kind of pull it in and then pushes it out the other side. All right. Alrighty. And then here we can see one of the neat things about this too. I talked about, alpha helices are very common to go across. We can also see beta sheets as well too. So you can actually make a beta barrel. To me, I look at this, it looks like, like the Chinese finger trap that, like the kids get for Halloween. So like put your fingers in there and you pull it and you can't, it tightens on around there. So we essentially make these pores out of beta barrels. We kind of weave in a little section in there. Okay. And multiporin has to allow maltose across, so it's fairly big. I also like to think of these as like the trash cans where you step on them and the lid raises. So something happens, you can open it up and close it. You can sort of see this might be our lid up here. If we change the conformation, we block, we close off the pore, we, change the conformation a different way, we open it up so we can allow something in. More on that type of mechanism a little bit later on. Okay. So again, this is going back to what I mentioned here. Just another way to look at this, we have selectively placed nonpolar amino acids within the membrane. Those are usually playing some significant role. Okay. So we'll see that here. We might have, say, a transmembrane region. We might see a particular charged residue in there. There is a neat way to try and figure out, is your protein integral or transmembrane? You can do what's called a hydropathy plot, where you kind of look at the sequence and plot its hydrophobic or hydrophilic nature. So we can see in this first one here, which is glycophorin, we have this one particular region here that's shown in yellow that is very hydrophobic. So there is probably a single transmembrane section there. Maybe that's an alpha helix that goes across. Who knows? We look at bacteria rhodopsin, which we actually kind of saw up here. Where is it? Right here. I believe this is bacteria rhodopsin. All right. We can see that we have, 7 different regions that are hydrophobic. So these are going to be those 7 alpha helices that kind of wind back in and out. And you can see hydrophilic characteristics in here between the two. Some of these it just turns right back around and goes back in there. But our hydropathy plot can tell us, if we have a hydrophobic region, that's probably going to be a transmembrane region of the protein. Okay. Something real quick here. Make sure I'm not dragging you guys out way too long, but we can do a little bit more today. So, we know that now that our membranes are made up of lipids and proteins and occasional carbohydrate, modifications in there. So let's talk a little bit more about the actual properties of the membranes themselves. They're very dynamic. We can see a lot of change that happens. The stuff that's in them is constantly moving around as well too. So these are also flexible structures. They can exist in different phases and undergo phase transitions. It's like, what? Solid, liquid? Well, we can talk about that. A kind of a more gel-like and a more liquid-like composition in here. They function as semi-permeable membranes, so they're not permeable to large polar solutes and ions. They are permeable to small polar solutes and nonpolar compounds. Those can freely diffuse across. And then we can affect the permeability by, treating it with chemicals in here. So if we want to get DNA into the cell, I know I mentioned electroporation. I used not an electric shock, but a high concentration shock to get my plasmids to transfect it into bacteria when I was working with them. All right. So let's go back to these phases. We can have a gel-like phase and a fluid phase. And our gel-like phase is more ordered. We're a little more solid. You think of like your gel inserts into your shoe or something like that. It's cushiony and squishy, but it's fairly solid-ish. Okay. So the individual molecules don't do a lot of moving around. On the other hand, if you think of something like motor oil or molasses, well, it's a liquid. Okay, it's a really thick, viscous liquid, but it moves around. So we see a lot of movement in there. Okay. If we heat up a membrane, we can cause this phase transition from this gel phase to the fluid phase. And if we cool it down, we can reverse that. Okay. Typically, under physiological conditions, we are a little bit more prone to the fluid phase than the gel phase. We got to be fluid so we can allow things to move through there, so we can adapt. Okay, so here we would have our gel phase. Things are fairly, as we draw our picture, our lipid bilayer, kind of everything's kind of touching like this, but really we're a little more commingled, like in the bottom picture, the liquid disordered state or the fluid phase. All right. We can also see, and some of you may have done this in various labs in the past, where you looked at maybe E. coli and heat shock proteins. If we grow E. coli in a very too warm conditions for them, we'll see heat shock proteins are made, which will help stabilize. They'll enhance the gel phase and enhance the stability of the membrane as a whole. Okay. But we can adjust our membrane fluidity by the glycerophospholipids that make up the membrane. Okay, so more fluids. Okay, um, gel-like ones are going to have longer saturated fatty acids in here because they will pack together more tightly and nicely. So we'll see this melting temperature will decrease as we add double bonds. We throw in those kind of kinks in there, trying not to flip everybody off, by biting my fingers, but they can't pack together as nice and neatly as if we have straight saturated chains. Okay, so that will decrease with our increasing number of double bonds. And of course, the longer, can't make my fingers longer here. My fingers and my toes. Okay, no. But the longer our chains are, the the tighter they can pack together as well too. If we have shorter ones, then our hydrophobic head regions can dominate there as well too. So we will increase that melting temperature as we increase the length of the saturated fatty acids. Okay. At high temperature, we tend to see cells will have more long saturated fatty acids in there. At lower temperatures, we have more unsaturated and shorter fatty acids. Okay, so just kind of look at this. If we take E. coli and start growing it up, we can look at the percentage of myristic acid. Myristic is, I believe, fairly short here. What we got here? I can't remember off the top of my head here. But if we look at a ratio of unsaturated to saturated, we can see that our unsaturated fatty acids, at lower temperatures, are fairly high percentage, not 2 to 3%, maybe not that high, but as we increase the temperature, we get fewer and fewer unsaturated fatty acids in here. So at 40 degrees, it's like less than 0.4%. Okay, we can also see that our oleic acid decreases, our palmitic acid actually increases because it's longer. Okay, I think myristic acid is maybe like 12 um, carbons. I'm not, can't remember hydroxymyristic. Yeah, but anyway, we see this change. We also see here, our shorter oleic acid dominates at 10 degrees and is greatly reduced at 40 degrees. Okay, so we can change our composition of our membranes to allow them to maintain their functionality at different temperatures. Okay. All right. We can also have our various sterols, our cholesterol, ergosterol, hopanols, various things can increase the membrane rigidity and permeability in here. We can kind of chunk up our membranes as well too. All right. So that's where we are. All right. Kind of looking at where we are here. I think we got a few more minutes. We can do some more if if my child actually does his work in the other room like he's supposed to. But anyway, we can affect our membrane rigidity and permeability by adding in cholesterols. All right. I always kind of think of it as like it's like making a soup. We're gonna throw potatoes in there to thicken it up or something. Yeah, cornstarch maybe. Anywho, so hopefully that's not too bad of a description of our membranes. Okay, we got lots of different components. We can kind of figure out what those components are, but our different components help regulate our functionality and our dynamics of our membranes. So if we need them to be more fluid, we can change the different components we're building them with. Okay, the last thing I want to talk about today, I don't know if you guys can hear the vacuum in the background, but I'm like, oh, really? Maybe I should just go to Starbucks or something and try and give my lecture, hiding. I went to Brook today. I couldn't hear the guy on the phone because it was too loud with the air running and somebody cleaned that. There's no quiet places. Gonna put like a little Wi-Fi extender router out in my shed and just sit on my lawnmower and teach from there. You guys can watch the bees fly around behind me. All right. Anyway, the last thing I want to talk about today, fortunately, are diffusion through the membrane. So we're going to talk about both lateral and transverse diffusion. And lateral diffusion is we just take a phospholipid and move it sideways. Okay, so it's staying on its same half of the bilayer. It's just moving sideways, and this is really fast. So we can just take this little membrane and scoot around all over the place there, okay? Um, it doesn't need to be particularly catalyzed, okay? How do we know this happens? Well, there's this neat study called fluorescence recovery after photobleaching, or FRAP, which is just really fun to say. It's like FRAP, okay? Um, so we can monitor this diffusion by looking at the rate of fluorescence return. If we tag our phospholipid head groups with a fluorescent molecule and then zap that with a laser to kill the fluorescence, we can measure how fast fluorescence returns as unzapped molecules diffuse into that space. Okay, and what we've determined from this is that you could take a lipid, it can go all the way around the membrane of an E. coli cell in 1 second. So it's like, woo, yay, I'm fast. Okay, but let's take a look at FRAP and how it works. Okay, this is a big overview. We actually have the next few slides are kind of breaking it down in here, but here we have our cell with our lipid bilayer. We're going to tag the external lipids with a fluorescent probe. It'll bind to those lipids on the outside. Okay, so now we're going to take a look at this under a fluorescence microscope. We can see, yay, everything's glowing there. So then we take a laser and zap this little section that we're looking at, and we make this hole, okay? And then in this hole, we measure how fast lipids come back into it. That gives us the rate of fluorescence return. That's going to be proportional to the rate of diffusion, of lateral diffusion of our phospholipids, okay? So bigger pictures in here. Here's our cell. Here we treat it with our fluorescent probe. Okay, so now the entire outside is treated with a fluorescent molecule. We take a look at some little section in here. We got that section there. We zap it with a laser that will kind of destroy our probe. So now we have this one little section in here that doesn't have fluorescent probe on top of it. Okay, so we monitor the fluorescence. We quantify the rate of fluorescence return as phospholipids diffuse through there. They're going to move back into this blank area. We can measure how fastly that fluorescence returns, and that will correspond to how quickly our phospholipids diffuse through, I guess I should say laterally transfuse through the membrane. Okay. Now here's the neat thing. If we keep track of one particular lipid, okay, it might start over here, and you'll see it's going to stay in this region, and it's going to stay in this one particular region for a while, and then it kind of hops over here and gets stuck in another region, and it bounces around here for a while, and then it jumps over here into the green region, okay? And then after a while, it jumps over here. So we can say it's kind of like neighborhoods. We generally are staying in a particular neighborhood, and then we cross a particular street, or we go across a big thoroughfare or something like that, and we're in a different neighborhood. So it's not like we're truly moving randomly all the way around the surface of the cell. There are some constraints. There's some little regions in there. Okay. All right. Yeah, and I think that's a good stopping place. What I'll do on Tuesday is we'll probably do a little bit of review for the exam, but we'll also pick up and we'll talk about, in this case, transverse diffusion. And now what we're going to do is we're going to take a glycerol phospholipid, say on the outside, and flip it over to the inside. And this, unlike lateral diffusion, where it just zips around sideways all over the place, flipping it all the way over is really slow, as opposed to a half-life of like a second, microseconds, milliseconds. The half-life for flipping one of these over is measured in days. Okay, so I'd ask you guys to think about why is it so difficult, so much slower to flip one over from, say, the inside to the outside or the outside to the inside, as opposed to going sideways. Okay, but I think that's a good question to leave you guys with. And we'll answer that question on Tuesday if you guys don't look it up and read on your own. Yay, before then. Okay. All righty. And with that, I'll ask you if you have any questions for me.
So the exam is on Thursday, correct?
Yes, a week from today. Yeah. And there's a study guide posted up there, so check that out. I'm making up the exam now, and like I say, you're probably looking at another 10 to 12, 10 to 15 multiple choice questions, and 5 to 8 essay questions. All right.
Is there any way we could get extra time? Because I be struggling.
I will change the time limits on these as well too. I've noticed that I can see how much time it takes everybody to finish, and when a lot of people are kind of bumping up against the time limit, I'll say okay, I'll expand it out there, and that's unfortunately that's kind of like the this being the first semester I'm teaching this class online. I'm like, oh, okay, and it's like how long should this take? So yeah, but I'll bump up the time frame for this exam.
Thank you.
You're welcome. Yeah. All right. One last reminder, make sure you find that little assignment for the survey about Biochem 2. That'll be open until tomorrow afternoon, something like 4 or 5, I think 5 o'clock tomorrow afternoon. But it's just like a one question thing of how likely you or how interested you are in Biochem 2. Other than that, I want everybody to have a good weekend. I guess I'll talk to most of you on, well, all of you on Friday for the lab. But yeah, that'll be very short and brief.